Now for the man who's leading research in India to find a potential vaccine. That man, the newsmaker, is now with us on this program. I would like to welcome Dr. Shekhar Mande at this moment. Dr. Mande, thank you so much for joining us right here on NDTV. My first question, CSIR, which is the nodal agency of research at this moment, is doing a lot of research on a drug called MW, a vaccine called MW. First of all, if you'd like to reiterate or tell our viewers what MW is in the context of COVID-19, where has the research reached?
MW is a bacterium. It's called Mycobacterium MW. The correct biological name is Mycobacterium indicus pranae, and it was discovered in India and has been tested very extensively by Cadila Pharmaceuticals. And they have also finished clinical trials on gram-negative sepsis, where they have shown that it protects about 50% mortality rate. It brings down the mortality rate in gram-negative sepsis by about 50%. What we have done that with the hypothesis that some of the other bacteria or other vaccine candidates which boost host immunity just like MW does. So we want to test the hypothesis that if MW boosts the host immunity, whether it will protect against corona or not. And that's why we have approached DCGI, and DCGI has given us kindly 3 different approvals for conducting the clinical trials.
Very nice. Very nice. Great to hear. Now, Dr. Mande, my next question is that, a lot of research the world over is happening. One such vaccine is the vaccine against Ebola virus. Beg your pardon. Now, I'm forgetting the name. I think it's Remdesivir, if I'm not mistaken. Would you like to tell us how far have we reached as far as Ebola virus vaccine is concerned in fighting COVID-19?
So the drug that you're talking of is called Remdesivir.
Yeah, Remdesivir.
And the global patent is on the name of a company called Gilead. They also have a patent in India. And they make a claim that Remdesivir is very effective against corona. And the claim, as far as the report in newspapers and media, seems to be pretty strong. So it will depend upon how Gilead markets it in India. And that will depend upon the strategy of Gilead to bring it to India.
My very simple question is this. Do we have any independent research to support that this Ebola virus vaccine will be equally compatible in fighting COVID-19? Any independent research?
No, not to the best of my knowledge.
See.
I don't think Remdesivir has been tried in clinical trials in India, not to the best of my knowledge.
I think there was a reason why I asked you that question, Dr. Mande. All right. Now, Dr. Mande, many countries have started what we call the human trial, and that this is considered a very significant phase. Where do we stand in our country as far as human trial are concerned on the various drugs that we are researching?
So the human trials, what we are studying on MW, are actually human trials. You know, that's a human trial on MW. That's exactly what we are studying. And similarly, other companies are gearing up now. As far as the drug domain, most people are focusing on repurposing drugs, because finding new drugs is a very long process. It takes about 8, 10, 12 years. But the repurposing drugs have already been shown to be safe for some other disorder or some other disease. And many companies they have reasons to believe that some of the repurposing drugs might be active against corona, and they're getting ready for it. The front runner, of course, as you said, is Remdesivir. But there are other drugs like Favipiravir, there are drugs like Lopinavir and Ritonavir, which WHO, through the solidarity trials, would like India to test them. And some companies in India are gearing up of conducting human trials on Favipiravir and all. I'm pretty sure that one of the companies in collaboration with CSIR
Dr.
has
Mande,
approached DCGI, at least for conducting trials on Favipiravir.
For Dr. Mande, for a better understanding at this moment, you're basically trying to say that human trial in our country is on as of this moment on a few drugs or a few vaccines. Can you give us more clarity? What all are the other drugs or other vaccines?
Okay. So the actual trial has not begun.
Ah.
We have received the approval from DCGI to conduct trial. We are partnering with 3 hospitals, and the process is that we have to take the institutional ethics committee approval before we start the trials. Everything has to be done with prior consent of the people who will receive a dose of this particular vaccine. What I'm given to understand is that many companies in India are also gearing up for vaccine trials. That's what I have read, newspaper reports and all. And they must have their own rationale on which vaccine to try first and which vaccine to try later.
Okay. Significant point. See, it was great to get a clarity on that point. Dr. Mande, my next question is, I believe that you have given some sort of a presentation in front of a group of ministers meeting chaired by the Health Minister, Dr. Harshvardhan. Would you like to share with us what you showed in that presentation?
So that is essentially about MW, what I spoke there when I presented my research, CSIR's research. And MW is the thing that CSIR is trying because it's an immune boosting vaccine. But I also presented about the repurposing drugs that I talked about. And what CSIR has done is CSIR has generated complete chemical synthesis route of the repurposing drugs. You know, what happens is that when the DCGI gives an approval to conduct trials, and let us assume that the trials are successful, at that stage, we should have an efficient synthesis process for our generic industry so that they can start producing that drug next day. And that's exactly what we have done. Our synthesis process has been worked out very in details, and we have handed over the synthesis process to a few companies so that if the trials are successful, these companies can start marketing that the next day.
Interesting. Now, Dr. Mande, this question I really wanted to ask. India has been kind enough to give hydroxychloroquine or HCQ to the whole world, to many countries in the world, because we produce it in abundance. But is it proven that it works? What does research say?
So we have not conducted any clinical study on our own, at least CSIR has not done it, on the efficacy of hydroxychloroquine.
Oh, so you're saying, that's short and sweet. So you're saying that there is no study on the efficacy of HCW. Wonder why the whole world wants it. My next question, Dr. Mande, is related to the Chinese testing kits. Now we know that they've proven to be unfit to be used. They are faulty. There is a lot of complaints. So my first question to you is, why do you think they failed?
So I don't know the reasons. I've read the reports in newspapers and on your channels, etc. And what I understand from those reports is that these were antibody testing kits. We have to remember one thing, that antibodies typically develop after about 6 or 8 or 10 days of infection. And in the first 6, 8 or 10 days, human beings may be walking around with virus infecting them, and they're essentially carriers of the virus and they can infect other people, but yet the IgG antibodies have not yet developed. So I'm not very sure why the kits have not worked, and we'll have to go into the details of that if those purported reports are to be correct.
Now, Dr. Mande, one question that has been asked, I'm sure you will judge me for asking this, but it must be asked because this is a conversation on the streets. There are studies that show that the antibody testing kits from China have a very low success rate, 5.4% to be precise. Why were they ordered in the first place?
Oh no, they have actually their own merit. I mean, we should not brush it completely under the carpet because antibody kit and the current gold standard RT-PCR, they have their own advantages. RT-PCR, of course, is the most confirmatory test as on today, is the gold standard in the market. But antibody tests are extremely important because there are many asymptomatic people who may be walking around and who at some point of time had been infected with the virus and they got cured on their own because their body's immunity fought very well with the virus, or those people who have recently become infected and have yet to develop symptoms of the disease. So both these kind of people, actually if they show antibodies in their body, they're actually very good people, because the hypothesis, and I emphasize it's a hypothesis, is that it would be believed that these people will not get a second infection very soon, and therefore these people can be deployed, for example, at the front in treating patients and things like that. But once again, there's a hypothesis, and we need to wait for whether second infection occurs or not.
Okay, okay. Now, Dr. Mande, before I let you go, my final question to you. See, we are importing these kits right now, testing kits. How soon before we start manufacturing our own Indian manufactured kits, testing kits? Where have we reached in that research? Because I believe there's a company, there is an institute called SCTIMST in Trivandrum that is doing great work. Any update on that?
So that's one thing where India has actually made significant progress, and it was to be expected. If you had asked me this question about 1.5 months ago, I would have told you exactly the same thing, that what we expect to happen earliest are the diagnosis kits. There are two very promising candidates around the corner. The SCTIMST, the Sri Chitra Thirumal Institute of Medical Science and Technology, they have a wonderful group which works at the interface of both the medical sciences and the engineering sciences. And they have come up with a test called RT-LAMP. This is essentially some sort of a PCR. But in typical PCR, you keep changing the temperature between 95, 72, and 55. And you go through the cycles of this temperature about 25, 30 times. Now, in the LAMP test, you don't need to change the temperature again and again. And there are multiple primers which anneal with the virus RNA, and at constant temperature you can carry out this particular amplification, and therefore the test becomes very rapid. Moreover, because it's not a quantitative RT-PCR, the machine does not cost as much. So the test is cheap, the test is fast, and the test is also accurate. The second important development in the country that has happened is the paper-based diagnosis that CSIR laboratory, Institute of Genomics and Integrative Biology, they have come up. So the young scientists there have used this enzyme called CRISPR-Cas, and they have coated the RNA and antibody on a paper strip, just like a pregnancy kit. And this test also is very rapid, very fast, and very accurate. So both these are, I believe, are very exciting developments, and that would be India's contribution to global science.
Oh, fantastic, fantastic. I can't wait before they get proven and they're approved, and then first used here, and then perhaps we help the world with these kits. Thank you so much, Dr. Shekhar Mande, for joining us with all the latest.